Everyone for joining us on this month's MDA Advocacy Institute. For those of you who have joined us in the past, you know that this is a new time. We are doing, I think, our very first evening Advocacy Institute. In the past, we've done them during the day, but we know for everybody who otherwise has day jobs, that can be a challenge to join our webinar. So we wanted to schedule one for the evening for those of you who perhaps haven't been able to join us during the day. And so for all of us, all of you who are spending your Thursday evening with us, if you're on the East Coast at the very least, we're really grateful for you joining us this evening. So I'm Paul Melmeyer, and I am MDA's Vice President of Public Policy and Advocacy, where I lead our public policy and advocacy efforts. And today's topic is on unlocking treatment development for rare neuromuscular diseases. And we wanted to cover this topic today because within neuromuscular diseases over the last 10 to 15 years or so at this point, there's actually been quite the advancement in therapeutic development, both preclinically, going into clinical trials, and actually from there, new treatments being approved by the Food and Drug Administration. We're actually nearing 20 treatments approved by the FDA for neuromuscular conditions, and that really is fantastic. But on the other hand, these of course are mostly in just a handful of diseases. For example, spinal muscular atrophy has several treatments now approved, as does Pompe disease, as does ALS, though of course we know with those therapies, they may not be particularly satisfactory to all, as with Duchenne muscular dystrophy. But there are many other neuromuscular diseases that still actually have no treatment approved whatsoever by the Food and Drug Administration. So we wanted to spend today talking about why we see some differences between certain diseases, either within or outside of neuromuscular diseases for that matter, having lots of exciting treatment development and lots of new drugs being approved by the FDA. And then in some areas, other areas, we just don't quite see that yet. Perhaps there's some clinical trials ongoing. Perhaps there are some drugs that are nearing the finish line, but still, when you look at these diseases compared to others, they're still not quite caught up to some of other neuromuscular diseases or other rare diseases for that matter. So we wanted to spend today talking about how, for especially those diseases, how we can unlock treatment development in rare neuromuscular diseases. I am joined by two absolutely fantastic colleagues, and that would be Ron Bartek of the Friedreich Ataxia Research Alliance. Ron might not be a stranger to many of you, as he, of course, is active in just about everything there is to be active in rare diseases. He's on the board of the National Organization for Rare Disorders. He has served with the Alliance for Stronger FDA, with the Alliance for Regenerative Medicine, and positions at the NIH and at the FDA. If there's a way in which Ron is able to contribute his extensive knowledge in rare diseases, Ron, you're doing it, and I admire that. So I am very, very grateful. We are all very grateful for you spending your evening with us tonight.
Way too generous. Thank you so much, Paul. Pleasure to be with you and Angela and our audience.
Fantastic. And then, of course, we're also joined by Angela Leck, who is the Vice President of Research here at the Muscular Dystrophy Association. Angela, you've been with us just for a little while now, but of course, Angela, you've spent several years now within neuromuscular diseases, serving, of course, doing your postdoctoral fellowship under Dr. Kunkel, one of the forefathers of neuromuscular disease research, of course, and now, of course, lending your expertise with us here at MDA and really guiding our research portfolio and really doing everything that we can do in trying to accelerate research in neuromuscular diseases. So Dr. Leck, really grateful to have you with us this evening as well.
Hi, everyone. Thanks for having me.
Okay, so let's actually go back one slide. I apologize, Mark. Let's talk about what we're talking about today. So as we've already gotten introductions out of the way, let me also do a few housekeeping items. If you have questions for us, we're going to be answering those questions both as we go through our discussion this evening, as well as at the end. If there are questions that are really pertinent, really kind of on the, on the nose for the topic that we're discussing, we'll turn to those questions as we're going along. And then for other questions that are also very important, we'll answer those at the Q&A section that we have scheduled for the end of our hour together. For you to ask questions, just go down to the Q&A box at the bottom of your screen there, type your questions in there, and our panelists here can be able to see them, and we'll be able to answer them if we're able to get to them, certainly. But the topics that we are going to be discussing today, you can see in front of you here. We're going to kind of start at the beginning, in a sense, of the research ecosystem, which is with basic and translational research, and then move into some of those policies and programs that we know to be very influential in actually leading to successful rare neuromuscular disease drug development. We're going to be covering the economic incentives that actually push companies, the biopharmaceutical companies, towards developing rare neuromuscular disease treatments. We'll talk about the importance of natural history registries as they pertain to better understanding rare neuromuscular diseases and actually facilitating drug development. We'll talk about clinical trial structures and how the success of a clinical trial really can actually turn on the way it's been set up, on the structure of that trial. More so than anything else, unfortunately, at times, it's the structure that actually will lead to whether that trial is to be successful or not. We'll talk about some programs FDA has in place to try to speed therapeutic development and also then speed regulatory review of these therapies from there. Talk a little bit about how the review divisions at FDA actually approach rare disease therapies, and in particular rare neuromuscular disease therapies, and how the review division is set up, and also the attitudes and approaches it takes towards rare neuromuscular diseases can be quite impactful in successful therapeutic development. And then finally, even after all of this has happened, once that therapy is ready to actually be on the market, it's been approved by FDA, how the perspective of the biopharmaceutical company on what the potential market there is for that treatment can be so impactful on actually it being developed in the first place. So we have a lot to cover. Let's get right to it, because we certainly want to be able to get right into the meat of our conversation here. So we want to start really at the beginning, and that's at the basic and translational research that we all hope is actually going to turn into treatments, a little ways down the road. So as we know, with some communities within rare neuromuscular diseases, the main reason for why therapies are not reaching patients as of yet is just simply because the science hasn't evolved far enough for these therapeutic interventions to actually be explored by the biopharmaceutical industry, and then from there being hopefully approved by the FDA. So Angela, as our resident research and scientific expert with us today, I wanted to ask you, why are some of these reasons for why the basic and translational research into some neuromuscular diseases has just not advanced far enough for treatments to actually be developed?
Okay, so now when you say the science hasn't evolved far enough, this can really be broken down into several issues. So firstly, most neuromuscular diseases have an underlying genetic cause, meaning there is a mutation in a certain gene that is either inherited or sometimes can arise spontaneously. And to know what gene and the type of mutation that is causing a particular disease is what's known as having a genetic diagnosis. Many neuromuscular disease patients have a clinical diagnosis, but they don't have a genetic diagnosis, meaning that the root cause of their disease on a genetic level is actually unknown. So it's really difficult to begin strategizing therapies for diseases without knowing the underlying cause of it. So I guess for a subset of patients, not being able to find out the root cause of their disease is definitely a huge barrier to be able to receiving therapy. But having said that, not all neuromuscular diseases have an underlying genetic cause, as you may know. So for a large portion of ALS patients, also for autoimmune disease patients such as myositis, for example, they don't have an underlying genetic cause. And for these, a major reason why there have not been therapeutic interventions is because I would say the disease mechanism has not been well studied or understood. Knowledge of molecular pathways that are affected in disease state are really key to strategizing therapies for a disease. And really only by understanding why the parts are broken can we begin to devise ideas on how to fix the problem. But this also applies to diseases with a genetic cause, as I mentioned earlier. And it's not enough to know the gene involved, but the mechanism in which the gene functions. So its characteristics, such as when the gene is switched on, is essential to know before attempting therapy. So, for example, gene replacement therapy is now proving to be a feasible therapeutic approach for some diseases, where it's known that reduction or absence of gene expression underlies the disease, for example, Duchenne muscular dystrophy. But we know from a lot of studies that this gene replacement therapy won't work for all genetic forms of neuromuscular diseases. For example, those linked to the expression of a toxic gene like FSHD. So a lot of what's known as basic science research really needs to be undertaken before a therapeutic approach can be strategized. And then one last thing, in addition, once the underlying gene and molecular pathways involved have been elucidated, there is then another hurdle to therapeutic intervention, and this involves recreating the disease in an animal model, such as a mouse or a zebrafish or a dog. And it's only with these models that we can begin to test potential therapies for efficacy and dosing regimens that may tell us whether a candidate therapy will or will not translate to humans in clinical trials. And so these steps I mentioned require years, if not decades, of research and a lot of funding, and could underscore some of the reasons why, as you say, the science has not evolved far enough along for a therapy to be available.
Oh, thank you, Angela. Yeah, that's the exact perfect answer there. And Ron, I want to ask you about how advocates and advocacy organizations can kind of influence this research process of trying to understand the disease. Because, you know, FARA, the Friedreich Ataxia Research Alliance, has really been at the forefront of partnering with the NIH, partnering with the biopharmaceutical industry, partnering with principal investigators, academic and other research institutions across the U.S. So what can advocates learn from FARA's experiences in really trying to shape the basic and translational research goals within Friedreich ataxia, and all of that with, of course, the goal of having a treatment development from there?
Well, thanks for that excellent question, Paul. And I will pose my answer by first emphasizing that it would be impossible for me to overemphasize the importance of the relationships you've referred to with the NIH and working closely with the National Institutes of Health. For us at FARA, those relationships became the absolute launch pad for our whole research effort and have been a real game changer for us from the beginning. In fact, even before we established FARA, we had the good fortune of meeting the NIH Neurological Institute's program officer. That would be of interest to a lot of people on this call. She was the program officer at NINDS for pediatric neurodegenerative diseases, which include Friedreich's ataxia, that I will henceforth just refer to as FA. This wonderful woman immediately invited us to breakfast at the NIH the following week on a Thursday morning that was to feature a meeting of the NINDS National Advisory Council in its open session. So we had breakfast with her. She took us upstairs to sit in on that advisory council meeting. And during those conversations with her, she actually encouraged us to establish a foundation. We hadn't done it yet. We didn't have to tell her that we had already made that decision, but she actually encouraged us. She said she'd done enough due diligence on us to know that we were well placed geographically and by inclination. We'd just gotten the diagnosis of our then 11-year-old son. She said, we really, we've got several thousand neurodegenerative diseases, and we don't have an organization for FA for two-way communications between the Institute and especially your community, especially your scientists. So we think you guys would be well suited to establish that, single disease communication system between us and your community. And just to paraphrase Humphrey Bogart's famous last line in Casablanca, that was the beginning of a beautiful relationship. We became immediately almost actively involved with that program director, leading to only 6 months later, after FARA had established, that FARA received a workshop grant, an R13 workshop grant that some of you might know about, that we used to fund the world's first international scientific conference on our disease. And it was co-hosted by FARA and NINDS. So that was really a seminal event for us. It really launched our whole collaborative research program with the 80 scientists that we were able to assemble for that two and a half day meeting. Later, I was asked to serve on that advisory council to which we'd been introduced. I finished a 4-year term on that advisory council, continued to serve on the executive committee planning the annual nonprofit forum for NINDS. Recently completed a 4-year assignment on the National Advisory Council of NCATS, the National Center for Advancing Translational Sciences at NIH, and co-chairing the Cures Acceleration Network Review Board that's closely associated with NCATS. The net result for FA basic and translational research, the topic of this question, is astounding. We enjoyed orders of magnitude increases in NIH funding for our basic scientists since we first got involved at the NIH. I think that was true clearly both because FARA's work with our FA scientists has better prepared them for submitting good applications, and because we've helped NIH understand FA and its most promising therapeutic avenues. They've often consulted us when they have questions about these applications. We've also gotten really good NIH help in translational tools that have included our collaboration with the Coriell Cell Repository to grow the availability of FA cells for research, our collaboration with the Jackson Lab for development of additional mouse models and characterizing those mouse models, and the NIH help for 3 of our industry partners. This help came from NCATS, in fact, in helping them complete their preclinical work so they could begin clinical trials. So I'd like to conclude by encouraging everybody to consider the various institutes at the NIH. There are about 27 of them, institutes and centers. For example, the ones I've mentioned include the Neurological Institute, NINDS, and NCATS, but also the National Heart, Lung, and Blood Institute. I know a good number of people on the call probably are interested in, to get to know them and to build relationships with their key staff people and participate in open sessions of their advisory council. All these centers and institutes have a National Advisory Council that consists of about 20 people, mostly the leading scientists in that arena, but also 4 to 6 patient advocates like ourselves. And you can participate live in those National Advisory Council meetings. Right now, they're all virtual, of course, but when we get back to in-face meetings, face-to-face meetings, you can go to these meetings, and give you a wonderful opportunity to meet the staff, to have lunch with them, to take them to dinner, whatever, to just get to know, become familiar with their programs, with them. And I would also encourage you to become familiar with NIH funding opportunities, and communicate those opportunities to your scientific investigators. And when they are ready to apply for NIH grants, you could even offer letters of support from your organization. So I'll conclude that now. Great question. And we couldn't be more thrilled with the kinds of relationships we've been able to build, and encourage everyone to do the same.
Very well said, Ron. And truly your experience in navigating all of these relationships and institutes and organizations and whoever it happens to be is is not only, of course, bearing fruit for the FA community, but really can be quite the lesson for everybody in other communities who are looking to shape that same kind of research ecosystem that you were able to shape through these efforts. So I want to move on to our next topic, and that's on the economic incentives that actually, if that basic and translational research has resulted in something that is encouraging to be a potential new therapy for the neuromuscular disease in question, what are those economic incentives that are in place that actually make doing this clinical development at a biopharmaceutical company actually appealing? Because as we know, these are rare diseases. These are diseases that they're all rare, and some are incredibly rare. And it might not make a lot of sense for a biopharmaceutical company to pursue this rare neuromuscular disease rather than that 30th drug for high cholesterol, for example. So there are already in place some incentives that try to push the biopharmaceutical industry towards rare diseases. These include some of the incentives that are in the Orphan Drug Act, which is actually the very first law that tried to and has been very successful in pushing companies towards rare diseases. For example, within the Orphan Drug Act, companies allowed to have a longer market exclusivity period to sell their rare disease drug compared to a common disease drug. So that allows them to sell it for longer without competition coming into the marketplace from a generic manufacturer, for example. There's also tax credits. At this point, it's a 25% tax credit that's available to a rare disease drug company that they're allowed to take on their manufacturing costs, as well as grants programs that were created at FDA to help for FDA to actually infuse dollars into rare disease research, and then also some exemptions from fees that companies would otherwise have to pay FDA as they are going through the regulatory process. Rare disease companies don't actually have to pay those fees. That's from the Orphan Drug Act, but ever since then, there actually are some additional incentives that have put in place as well, such as rare pediatric disease priority review vouchers. If a company develops a drug for a rare pediatric disease, they get a voucher from the FDA that they can either then gain a priority review, and we're going to be touching on priority review later, don't you worry. Priority review for another of their drugs, or they can sell that to another company. And some of these companies have made a lot of money from that sale, which again pushes these companies towards rare diseases and rare neuromuscular diseases as well. So Ron, I want to start with you for a question on incentives within rare disease drug development. Can you tell us how these incentives have been important to developing treatments for rare neuromuscular diseases like Friedreich's ataxia, like FA?
Mhm.
And it might not be intuitive to many for why advocates should be advocating for or even concerned about financial incentives in drug development. Like that doesn't perhaps ring true to many, but why is it so important for advocacy organizations and advocates to be involved in that conversation?
Well, great questions again, and I'd say the answer to both those questions is the same. We're absolutely convinced at FARA that there are two ways in which these incentives have been extremely important to us and to everybody in the rare disease community. First and most obviously, they've clearly helped pharma companies turn their attention to rare diseases generally. It wasn't that long ago when we first got involved, for example, of course, that was now 22 years ago, that it was just the beginning of this movement to pay any attention at all to rare diseases. Everybody, said, and they said to us initially, you're too rare, you're on your own. They weren't interested. But that has changed drastically and in large measure because of these incentives. So that these incentives are helping pharma companies generally turn to rare diseases and a focus when otherwise they would be focused, as you said, on much larger markets. And the second reason is they help these companies become interested in our particular rare diseases. If those companies believe that our organizations have the basic and translational assets to help them get their very best shot on goal in developing their therapeutics, and that our organizations can help them inform their applications for some of those incentives. For example, the one that Paul mentioned, a rare pediatric disease priority review voucher, if they're convinced that your organization has all the assets they need to inform their clinical trial design, to inform their endpoint selection, to inform everything about their clinical research, and then also provide them help on applying for these additional incentives. They will come not only to the rare disease community, but they will come to your rare disease. So we're convinced that these incentives have been important in drawing hundreds of additional companies to rare disease generally, and combined with the basic and translational tools we've developed, those incentives have grown the number of FARA's active industry partners from 0, which is what we had at the outset, to about 4 dozen right now. And these are very active industry partners for whom we've helped a number of them apply successfully for example, the rare pediatric disease designation. So in those 2 ways, drawing their attention to rare diseases generally and to our various organizations can help draw their attention to our disease group in particular. That's how those incentives have helped us.
Yeah, very well said, Ron. Thank you. Dr. Leck, I want to ask you about the intersection of these incentives and especially complex treatments. So we're talking about gene therapies, cell-based therapies. I mean, you talked about finding the genetic underpinnings of neuromuscular diseases and the advent of genetic medicine within neuromuscular diseases, for that matter. These are incredibly complex therapies, though, that would kind of stretch the bounds of our current system. What would you have to say about whether the incentives in place right now for biopharmaceutical companies to be developing therapies are keeping up with the complexity of genetic medicine and neuromuscular diseases?
Yeah, so that's a really big challenge in the field right now. The current incentives are really not adequate, particularly when you're talking about these one-off, really expensive personalized therapies. So also when biotech companies, when they say they're interested in rare diseases, they also often mean the most common of the rare diseases, such as your Duchenne muscular dystrophy, myotonic dystrophy. So disease prevalence really plays a huge role in how attractive a disease is to work on, because it defines how big their target market is. So with gene therapies such as AAV-based gene replacement therapies, that you may have heard of, and it's like quite the big deal right now. It's a one-shot deal, meaning, a patient can't be redosed. For some ultra-rare diseases, this will mean that there will be no more patients to treat outside of the clinical trial. So how can a company expect to make a profit if all their patients have been treated in just a single trial? You can do the math and see why the incentives are just not in place for companies to invest in treatments such as gene therapy that costs $2 million per patient, not to mention the incentives for also insurance companies are also not in place to fork out that amount of money for a single therapy in a single patient. There's also, issues of incentivization for companies to pursue neuromuscular disease indications across the board instead of biasing the more also severe forms, because the milder forms are more difficult to show a therapeutic benefit in a short clinical trial window. And so these severe forms typically constitute the childhood onset neuromuscular diseases. So the later onset forms that are milder make for less attractive investment, despite still causing very severe debilitation over a long period of time. So there's a lot of work that needs to be really done in the framework to incentivize the companies to really start investing in gene and cell-based therapies for rare neuromuscular diseases.
Yeah, thank you, Angela. I think a perfect summation that I've heard from both of you is that the incentives are incredibly important, but imperfect. They don't perfectly incentivize every single area that we want them to, and there are certainly areas, as Dr. Leck has said, such as in one-time dose gene therapies, such as in some of the milder forms of some of these diseases, that the incentives just aren't adequately pushing companies in that direction right now. So I do want to move along, if we don't mind. We could talk about incentives all day, but we have a lot of other important topics to get to. And next up is natural history data registries, because they truly can be so important and impactful in facilitating not only a better understanding of the neuromuscular disease in question, but actually also accelerating participation of communities within the clinical trials themselves. Dr. Leck, I want to start with you as we talk about natural history data registries, because of course, MDA has our neuromuscular disease observational research hub, which is serving, of course, as a natural history data registry. But Angela, I want to ask you about how these registries actually help us understand rare neuromuscular diseases and how can they contribute to drug development.
Okay, yeah, well, natural history data serves to track the progression of disease across a patient population, and it's really vital for several reasons. There's scientific reasons and also clinical reasons. Scientifically, tracking the progression of disease will really lead to a better understanding of the pathology, which in turn will enable better strategizing of therapeutic approaches. Sorry, that's my dog. Something's setting him off. Clinically, a clinical reason for natural history data is that it will help clinicians better manage disease in patients. So, for example, if natural history data points out that there's cardiac involvement that occurs at a particular stage of a disease, then the clinician can be aware and then take steps to proactively prescribe certain medications to treat these symptoms, because this natural history data is there, even though he or she as a clinician has never had another patient. They can be more informed in treating the current patient. And natural history data is also very important in the context of clinical trials. So firstly, natural history data will help guide your trial design by identifying the most appropriate patient cohort to target, and also pinpoint the relevant clinical endpoints. And in terms of clinical trials also, if a company has a potentially life-saving treatment, rather than having an untreated or like a placebo control group in a trial, where patients will be receiving, quote unquote, a sugar pill instead of receiving this potentially life-saving treatment, sometimes the FDA will allow natural history data to be used as a point of comparison for the untreated cohort. And this is important for patients because while you're on the placebo, you could be incurring irreversible damage that could have been otherwise treated by this life-saving drug. So having data on the natural history progression of a disease can inform you of your candidate, whether your candidate treatment has been successful and has altered the expected trajectory of a disease. And so the more patients in a registry, the more weighting the data will have, and the more likely the FDA will approve it as a point of comparison. So it's really important for patients to participate in natural history studies for their respective diseases if they want to help accelerate drug development and testing.
Yeah, very, very well said, Angela. And Ron, of course, there are lived examples in the context of FA that you and the organization and the community have seen as to the importance of natural history registries. Tell us about some of those and some of the ways in which other neuromuscular diseases could learn from the experience that you and the FA community have had.
Okay, Paul, I'd like to be very brief in answering that particular question, because if you still intend to ask the next question that closely relates to natural history, I'd like to expand on it there. But I'll say here, probably nothing more important to our community at all than the work we've been able to accomplish on natural history and our patient registry. I I name them both because we have two registries. One is a patient registry that includes demographic information on more than about 3,000 patients, and we primarily use that one for trial recruitment. And we also have our natural history database that houses data collected on more than 1,300 FA patients over almost 20 years. We've used that natural history database to almost fully characterize our disease and to provide our industry partners with what they consider an invaluable resource in designing clinical trials. And again, I'll expand on all that and how we use that natural history database and that patient registry in answering your next question.
Yeah, thank you, Ron. And we do have a question in the chat about registries. And Ron, I'm going to turn to this to you because I do believe you have some experience here. The question is from Josh about whether FDA has given any natural history study grants yet, as perhaps contemplated under the Orphan Drug Act, but it hasn't. Josh has yet to see FDA post any specific methods for applying. Ron, can you tell us if FDA actually is funding natural history registries yet?
I can tell you pointedly, because we have been one of the beneficiaries of such a grant. And this was a grant from the Office of Orphan Product Development we have this sort of natural history database that we're very proud of that is largely adults. Adults and older pediatrics. And we recognized that what we really needed to add is a natural history database on a younger and younger children with confirmed FA. And we applied for a grant at the Office of Orphan Product Development and one of our principal investigators, our clinical lead at Children's Hospital of Philadelphia, applied and obtained that grant. It's a substantial grant over 4 years to study the pediatric FA population. And we're closing in on the end of that 4-year period, and the data look remarkable. And we're just really thrilled with the way that this will help inform our further clinical studies in what we hope to be younger and younger patients, because their treatment window is so broad. So, compared to what it will be when they're more progressed. So yeah, the agency is providing such grants. I can speak only authoritatively to that one that we've been fortunate to receive. Absolutely.
And Josh, if you want to learn more about who to be contacting at FDA or what web pages to look for more information, please reach out to us, advocacy@mdausa.org. That is our email address to reach us. We can send you some information after today's webinar on more information on that, who to talk to and so forth, and exploring those grants. So Ron, you already teased our next topic, which is clinical trial structures. And I do want to turn to you first on this, because truly the structure of a trial can make all of the difference in whether that therapeutic development effort is actually going to be successful. If a trial chooses the wrong eligibility criteria, the wrong endpoint is selected, what otherwise actually may be a safe and effective treatment might not be able to show it in the clinical trial, and consequently, FDA may not be able to approve it because of that. So I know structuring a trial has been a challenge within FA that has been a challenge for years now. Tell us about your experience you've had within the community on choosing the right structure for a trial, choosing the right endpoint for a trial, and what you all have learned through trial by fire, I guess you could say, that could be of benefit to other rare neuromuscular disease communities.
Well said. I'd like to illustrate my answer to this very important question by outlining the typical course of events as our industry partners develop their clinical trials. And you'll recognize immediately this is totally unlike the old model with which way too many of us were completely familiar, about which we've all heard, where the companies came to advocacy organizations like ours only when they wanted help in recruiting their phase 2. I mean, they thought they had it all nailed and they wouldn't have to come to a patient group until they needed our help in recruiting a large, almost pivotal study. That's not the case with our industry partners anymore. It might have been in the first year of our experience, but as soon as we started building the assets that we were talking about here, a natural history database, patient registry, a clinical network, all of our potential partners started coming to us very early. In fact, some of the biggest partners came to us before they even had a candidate, before they had even a target in our disease, or even a therapeutic approach in mind. They came knowing that we had done several things. We had the assets they needed. We pretty fully characterized our disease with this natural history study going over, initially 10 years, then 15, and now 20 years. So we characterized that disease. They could look at any number of our publications and get the basic science that they would need to proceed. They saw too that we've defined the symptoms most important to our FA patients, so they can target their therapeutic to a symptom that's meaningful to our patients. And in this realm of patient-focused drug development, they've increasingly recognized how important that is to all of us. And they knew too that we and our key opinion leaders are ready and willing and able to share disease expertise and explain our natural history database to help them design their trials. For example, how to align inclusion-exclusion criteria to their specific therapeutic expectations, how to select endpoints most likely to demonstrate efficacy for that therapeutic approach, how to select subjects whom their therapeutic is most likely to benefit, and to select trial procedures most compatible with the capabilities of those subjects. And so what happens, they come to us early and start doing all that. They will have 2 or 3 meetings with our KOLs and us and our lead clinicians and basic scientists. And then they go back and start preparing their protocol. They then vet that draft protocol with us before finalizing it. And then they're ready to give that at least a protocol summary to us so that we can use our global patient registry, the one that features largely demographics, to which we can send a recruitment notice for their trial with the basics of their trial and what it will require of our patients. And we can send that to a targeted list of patients that seem to be, according to the patient registry data, most eligible to meet those inclusion and exclusion criteria and closest to the clinical trial sites that have been selected and so forth. So the result is clear. We recruit very quickly. We recruited, for example, I know a story I've told you before, Paul. We recruited a 100-patient phase 3 clinical trial at 3 sites across the country in a matter of a few hours. The PI started calling and said, stop. We've got all the patients we need in the waiting list. And that was in about 3 hours' time. We recruit very effectively with very few, if any, screen failures, very few, if any, dropouts or data loss, and no amendments. So the trial is completed on time with excellent compliance to the protocol. And in sum, we give our industry partners their very best shot on goal. We believe and can't wait for some of those shots to find the back of the net very soon. Final quick note, but important one about natural history and endpoint selection. Some of you have probably heard about the exciting program funded by the FDA and being implemented by the Critical Path Initiative with help from NORD. It's called the Rare Disease Cures Accelerator Data Analytics Platform. It was launched in 2018 and has been assembling fully conformed natural history data provided by our advocacy organizations and clinical trial data provided by our industry partners on as many rare diseases as choose to participate, and they then help analyze those data to facilitate endpoint selection and other aspects of clinical trial design to share lessons learned with all disease groups, especially those facing related issues. So once all these data are assembled, they're already helping us analyze those data. And they're also looking for ways they can share those data points and those lessons with other disease groups that have similar challenges with similar tissues and similar disease mechanisms. I'll just be braggadocious here for a second and say that FARA was the very first disease organization to submit to this program all of our natural history data and a growing number of clinical trial data sets from our industry partners. And our analysis being done in collaboration with that program is already producing really valuable insights about endpoints and that will be most sensitive to change in our patients. Just this afternoon, for example, we had a 1-hour meeting with our CPATH colleagues that are working on this program and key FDA leadership. I mean, the directors of review divisions and offices to review the insights we've already gained from this program and to plot next steps. So I really strongly encourage everybody in this meeting to consider how your disease group might become engaged with this rare disease Cures Accelerator data analytics platform and submit your natural history data that will be fully conformed to common data elements and so forth. And they'll add to those data and they will help you analyze it so you reach effective endpoints and clinical trial design. So I'll stop there.
Thank you, Ron. So, Angela, I want to ask you a similar question, and I actually want to ask if you could combine, in a sense, answer to this and one more, because we are running behind. But, you know, talking about the complexity of clinical trials and the challenges in showing safety and especially effectiveness in heterogeneous, slowly progressive neuromuscular diseases. Tell us about why choosing the right endpoint, the right structure of the trial is so important, especially in these heterogeneous and slowly progressive neuromuscular diseases. And if it also at the same time, you could tell us how surrogate endpoints can be particularly important in these kinds of trials.
Yeah, well, yeah, selecting the right outcome measures for a clinical trial is super important because it'll determine the success of your trial. It's the readout for whether a given therapy, you know, has shown efficacy. So outcome measures are basically selected measurements for those of you who aren't aware of the term that's been anticipated to show basically a significant change given an effective drug. So examples are like how long someone takes to get up from a sitting position or how long someone can walk in a given time frame. And these measurements, they're normally informed by natural history studies, as Ron mentioned, and which, you know, marks the progression of a particular disease and then can help to guide in selection criteria of patients who are most likely to exhibit a response to a therapy as reflected by improvements in specific clinical endpoints. If for whatever reason the endpoint measure selected for a trial was not informative as expected and does not show evidence of a significant difference between treated and untreated cohorts, then this trial is considered a failure and the drug will subsequently receive negative media coverage, investors will pull out, and then there'll be no more funds to conduct follow-up trials. So you can see that if you have a drug that works, you need to structure a clinical trial to accurately reflect that. You need to show it, and you pretty much get one shot at it. And the success of a clinical trial for a given drug, which is what will allow it to progress into the next trial or obtain FDA approval, really all hinges on selecting the right eligibility criteria of participants who have potential to show the most improvement in selected clinical trial outcomes. So if these parameters were wrongly selected, then a potentially effective drug, you know, it's scary that it may not see the light of day. And you also wanted me to touch on surrogate endpoints. They're super important because particularly for slower progressing in neuromuscular diseases, because clinical benefit is not very easy to demonstrate in many neuromuscular diseases where the aim is to simply stop the progression of the disease. So many candidate treatments in the space really rely on surrogate biomarkers being used as endpoints, such as, you know, increased expression of protein X or reduced transcript expression of gene Y because it's a related disease biomarker. And these surrogate endpoints, are what's been derived from very robust scientific studies that have established a relationship between certain measures and a disease state. Without relying on surrogate markers, it's really difficult to evaluate therapeutic efficacy of drugs in trials, particularly for adult onset neuromuscular diseases that are slower in progression, such as your limb-girdles, FSHD, GNE myopathy. Because for these diseases, the current drugs in development aim to stop the progression of the disease, not to reverse them. So it may take longer to establish clinical benefit, and that's where surrogate markers, particularly used in accelerated approval pathways, are very important.
Yeah, that's exactly it, Dr. Leck. Thank you. And since we are nearing our hour, I think we'll have to limit our conversation on these expedited approval pathways. Dr. Leck has already mentioned one of the very important ones, and that's accelerated approval, in which a therapy is actually approved based upon the performance of either an intermediate efficacy endpoint or a surrogate endpoint, as Dr. Leck has already described. But there are also 3 other programs that aim to actually accelerate the development of a therapy. Those include the Fast Track program, the Breakthrough Designation program, and the RMAT program, Regenerative Medicines Advanced Therapeutics designation program. Each of these are ways in which FDA can invest, in a sense, their time and attention towards trying to get these innovative treatments through a development process. And if they are particularly exciting, once they reach that end stage, FDA can also use a 4th program, which is priority review, which means they will review it that much more quickly than they would otherwise. Those 4 programs I just mentioned, plus accelerated approval, 5, are the way in which FDA tries to expedite drug development and review through these mechanisms. So I want to just ask one more question of Mr. Bartek, and that's the importance of developing relationships with FDA review divisions, and also just how FDA review divisions can actually impact how a therapy can be evaluated, whether it's the flexibility mechanisms they want to use, whether it be the amount of data they want to see. Ron, you have lots of experience in obviously developing relationships everywhere, as you talked about, but especially at these review divisions that are tasked with reviewing the safety and effectiveness of these therapies. Tell us what we can learn from how these review divisions approach these things differently, and how advocates can work with these review divisions to be sure that they're in a good place.
I love that question. I'll try to be brief, and I'll try to do it without emotion. This is a tough one for me because I'm so thrilled with the relationships we've been able to build over time with these people at the FDA. I always encourage our own organization and anybody else who will listen that if you want to accomplish something at the FDA, it's far more effective to try to do that with them rather than try to do it to them. And we're thrilled with the results of that fact in our experience, in which we've been able to build and nurture wonderful relationships that are characterized by mutual respect, appreciation, and obvious commitment from both parties to our patients. This is true for review divisions, both at the Center for Drugs, especially the Office of Neuroscience, with whom we met this afternoon, and the Center for Biologics, where we have a very good relationship with the CBER director, Peter Marks, and the OTAT director, the Office of Tissues and Advanced Therapies director, Dr. Wilson Bryan. And that's the office that Dr. Leck and Paul and I have been referring to in terms of gene therapy. That's the office that will review all of our gene therapy and gene editing proposals. And I met Dr. Bryan at an MDA conference a few years ago. So our role with the FDA is first to educate. We've attended and participated in any number of pre-IND meetings at the invitation of the development sponsor. We've participated in special by request meetings, type C meetings, all in close collaboration with our industry partners. And we've in doing so, have helped change the divisions and the industry partners' attitudes and expectations regarding how participation by patients and advocates can be helpful rather than disruptive. Both the industry partners and the divisions view our participation as expressing essential, positive patient voices. Note about an important element in all that is the patient-focused drug development commitment on the part of the agency and all of us. We participated in all the initial development meetings on PFDD back in 2012 before the legislation requiring it was passed. We've conducted our own externally led patient-focused drug development meeting, had very large FDA participation. The FDA keynote was delivered by the director of the Office of Tissues and Advanced Therapy, Dr. Wilson Bryan, who is a neurologist by training, by the way, and who told us, all the assembled hundreds of people, how an FA patient had changed his life. Final comments about this topic are that the FDA Review Division, now the Office of Neuroscience, was very involved in affecting our endpoints. Our primary endpoint is what we call the Modified Friedreich's Ataxia Rating Scale. We call it modified because of the input from the Review Division at CDER that helped us eliminate parts of it that were not very sensitive to change and expand on those that were. Final note is that we always are conscious of advocating before and for the agency. So we advocate before them for our patient community and our industry partners. We advocate for them by participating in annual efforts. For example, the Alliance for a Stronger FDA. The immediate past president of that organization that always advocates very effectively for getting the FDA the additional resources it needs to meet the unmet medical needs of our patients. So and I got through all that without getting emotional, at least obviously emotional. So it's a first.
Well, thank you. It's been quite the journey that you've been on with our friends at the Office of Neuroscience, and as well as OTAT, of course. Dr. Bryan, by the way, former MDA Care Center director, so always
cool.
a friend in a good place there at the Food and Drug Administration. So we have 2 minutes to the hour. Unfortunately, I don't think we're going to have time to cover our final topic, which was on the market for therapies once approved. Very important. Hopefully we can touch on that in the future Advocacy Institute. But before we close, there are a couple really good questions in the Q&A box that I'm hoping we can touch on. And the first is from Ryan. And this has to do at the intersection of FDA regulatory oversight and gene and cell-based therapy development. And he asks, given the molecular mechanisms of potential gene therapies, such as AAV or CRISPR, is it possible that regulatory frameworks could focus on the mechanism rather than the disease? If so, could a company develop the mechanism, receive orphan drug economic benefits, and still have a future market? So Ron, you are one of our regulatory experts here, but Angela, I'm going to turn to you first. Just to give a minute or two, if you can, on these kind of underlying platforms that could potentially treat many different rare diseases or neuromuscular diseases, and what Ryan means by the underlying mechanism that can then potentially impact many other rare diseases from there.
Yeah, so I think one of the really obviously attractive features of gene replacement therapy is that you can say, let's not worry about understanding the mechanism of how, this gene works. Let's just restore the correct functioning of the gene by putting in a healthy copy. And let's not worry about it trying to understand, every single thing the gene does. We'll just put the right copy back in. And that seems to be an accepted approach these days in terms of drug development, that not every detail needs to be figured out for a particular gene. And that so a lot of these gene therapies are starting to, I think what you're trying to get at is that it's becoming like a platform technology. where once a gene therapy has proven to be safe, you kind of just switch out different components or the different cassettes within the gene therapy itself. And so this can then lead to rapid approval of subsequent therapies for other rare diseases. So in other words, keeping the regulatory regions of the AAV as well as, other aspects, but just flipping out the gene depending on what disease you're trying to target. So getting approval for some of these gene therapies in SMA and DMD really has a positive impact and will lessen the time it'll take for other gene therapies to then subsequently be approved. So it does definitely pave a path for the rest of the other rarer diseases. Ron, did you want to continue?
Yeah, that's great. I'd like to just supplement all the fine things that Angela just spoke about in three ways, I guess. The first is that there are ongoing efforts that you might know about, Ryan, that combine the NIH and the FDA and our disease groups. And one of those is called PAVE-GT, which is a platform, as Angela indicated, a platform approach to very rare diseases. They've picked about 5 diseases in which they're trying to perform platform clinical trials using the same AAV9 vector, delivering the different genes, of course, for these 5 specific rare diseases. And the beauty of this too, Ryan, is that all the results will be published. All the results will be made commonly available. This isn't going to be something that these companies stick in their IP hip pockets and charge $100 million for. This is going to be all shared. The other program is collaboration between the NIH and the FDA. Dr. Peter Marks in the lead there on what's called Bespoke. It's a similar approach, taking additional rare diseases and advancing the mechanisms for advancing gene therapy in all of them and all other diseases. And a key element there is, talk about a process that's going to be the longest pole in the tent. It's manufacturing. That's been the big driver of big, huge costs. I mean, companies have spent I think the SMA gene therapy, Avexis spent like $200 million just on manufacturing for that program. And it was bricks and mortar. And we can't go on like that. And we can't go on charging millions of dollars per application. So anyway, there's all these attempts to consolidate and to share. And I'll say I don't think I've seen before the amount of pre-competitive sharing amongst companies and with the FDA and each other that we're experiencing right now in the AAV vector gene therapy community. So I'm really encouraged, Ryan, by all these developments, and I hope they all lead to the same end, which is more effective gene therapies for far more rare diseases than we have right now.
Thank you both. I want to just ask one more question because I think it's a really good one, and this comes from Stephen. And Stephen asks, this is in the ALS space, that there have been a number of natural history studies, but there has yet to be a successful effort to convince FDA to use natural history studies rather than a placebo in a clinical trial. So I think Stephen's asking, why is that? Why on some diseases is FDA more inclined to actually use natural history data as an external control, as a historical control, rather than a placebo group, whereas in other diseases, such as within ALS, they're not there yet. And I'll tell you what, I'll take moderator prerogative and start, and then I'll turn Ron and Angela to both of you for your thoughts. Generally what we have heard from the FDA as it pertains to why they are willing in some ways versus others, it has to do with both the understanding of the disease as well as the confidence the FDA has in whether the control of the natural history data registry will be as robust, I guess you could say, as the control that a placebo control group would provide. If FDA could feel that confidence that they're going to be able to discern safety and effectiveness from the experimental group with a historical control rather than a placebo control, then that's when they're more inclined to do so. But with an ALS right now, from what we have heard from them, they don't have that confidence yet.
Mm.
So that's what I would add. But Dr. Leck,
yeah, I want to add that I think ALS is very heterogeneous. I think the FDA, the scientific community, sees ALS as a very heterogeneous group of diseases or patients, given that there's not a single underlying genetic cause that combines them or that unites everyone in the ALS space. I mean, there's a few disease genes already associated with ALS, but for the majority of the ALS patients, like about 80%, the genetic cause is still unknown. So I think that the FDA can't feel confident that they're all kind of, even though they all fall under the same umbrella of ALS, that they could be slightly different diseases, but maybe manifesting in a similar phenotypic outcome, you know, that the root cause of it could be slightly different in all of them.
Yeah, Ron, what would you add to that?
Just quickly would add a couple points. One is, Angela just mentioned a really key in this one is it's the heterogeneity in which not just ALS is considered, but most of our diseases. I mean, even in FA, despite all of our natural history data, it's clear that not everyone has the same experience. And so it's really hard to convince the FDA to drop its insistence on placebo control because of that heterogeneity and, and difficulty in establishing, okay, what is the common experience here? So what we've experienced thus far is, no, we have not been allowed to use natural history as a substitute for placebo, but we've definitely used natural history very effectively to supplement the analysis of the placebo arm versus the active arm. So we can show with our natural history database that this placebo arm is functioning exactly the way our natural history functions. And so we can say, yeah, it's only a 2.5 point difference between placebo and active, but that 2.5 point is shared by all the other people out there in the natural history study. And here's how important it is that 2.5 points means 2 years of saved progression. So it's invaluable natural history data that's being put to good use, though not yet very broadly as a placebo control mechanism. So.
Yeah, thank you both. I think those answers are spot on. The key word here is heterogeneity, unfortunately, and unlocking the reasons for that so that we can understand precisely why that's the case. Ron, I'm just going to ask you one final FA specific question. This comes from Alicia. She asks, do you think there will be clinical trial for people who have had FA for a long period of time?
I can say definitely yes. And I'd say that there have already been a good number of clinical trials that include our beloved patients that have had FA for a long time. I would say even in gene therapy, I know I've had many conversations with Dr. Brian, and he always says, never give up on the later progressions. And so, yeah, it's one of the reasons they always insist that you start with adults. Yes, you need to establish safety in adults, but you also want to establish some efficacy for adults. And so, going forward, at least we will always insist that we make every effort to include older patients and in our clinical trials and in the benefits of these clinical trials. I mean, we want the label when we finally get an approval to include all our patients. And so we want to make sure we do the preclinical data collection and the clinical data collection we need to validate, verify that those labels need to include all of our patients, even those that have had our disease for a long time. So we have you in our hearts.
Thank you, Ron. That's a great way to close our session today. We have a final question from George, whether this would be recorded and posted. Yes, we have recorded this. We will be posting this on MDA's advocacy webpage. You can visit us at mda.org/advocacy. We'll have this up, let's say next week sometime. We're targeting next week sometime to have this recording live for those of you who want to go back and listen to some of our great insights again, or some friends or colleagues who weren't able to join us tonight. You can send along the recording to them so they can listen into all the amazing things that Ron and Angela had to say. So I will just close by, again, a point of gratitude. Ron, thank you so much for your time this evening. Dr. Leck, thank you so much for your time this evening. Very grateful to have shared the stage with you. Your insights are invaluable as always. And everyone, I hope you all have a fantastic evening.
Thank you. Have a good night.
Thanks all.
Bye-bye.